is the founder and CTO of Genialis, the RNA biomarker company. He's led the development of a pioneering biomarker based on RNA-seq and machine learning, which is accepted by the FDA for clinical trial use. He's passionate about data integration and XAI. Miha has also co-developed the Orange data mining suite and Resolvi bioinformatics engine. He earned his doctorate in computer science at the University of Ljubljana and completed postdoctoral research at Baylor College of Medicine. His talk is on explainable AI and precision oncology and how the, Zerner TME panel, an ML-based transcriptomic biomarker, earned the confidence of drug developers and regulators. And I apologize for murdering that.
Thank you. So I'm an AI geek by heart, but I'm also someone who cares deeply about giving back to the community. I was a scout leader, I contribute to open source and open science, and my work at Genialis is really all about one thing, which is helping patients get to the best outcomes. I have the privilege of working with some talented individuals. We have 14 PhDs on our team, two of them are MD PhDs with deep expertise in healthcare and machine learning. We are also a founding member of Alliance of Artificial Intelligence in Healthcare, which is an advocacy group that promotes responsible use of AI in medicine. So I'm happy to invite you to the panel in the afternoon when we'll talk about that. To set the stage, biomarkers, the way I define them, or we define them, are measurable indicators of a biological state or condition, and they're particularly useful for precision medicine because they can tell us about the disease phenotype from which we can guide treatment decisions. You have different kinds of biomarkers. Examples here would include you go to a doctor, you're given a prognosis, right, based on the prognostic marker. What I'm going to talk about today are predictive biomarkers. Here's where we focus as a company so that we predict patient response and patient selection for clinical trials. Our biomarkers process RNA data. That's why we call ourselves the RNA biomarker company. Standard of care today, particularly in cancer treatment, relies on single analyte biomarkers like the presence or absence of a mutation or gene. We, in contrast, develop complex markers of human biology that better define the phenotypes of disease, and then we link these phenotypes to specific therapies. Here we have two examples. To the left, for KRAS inhibitor treatment, the treatment is assigned based on the presence of G12C mutation in the KRAS gene. What we did is we modeled the pathway upstream and downstream to better capture the relevant biology. To the right, you see our model of tumor microenvironment that captures the interaction between angiogenesis and immune response. And I'm going to talk about both of these models that are built on RNA-seq and machine learning in two of the use cases. We model cancer biology through hallmark signatures. By signatures here, I mean predictors that capture some key aspect of tumor behavior. These signatures, in our case, would use RNA, which tells us about the tumor functional state at a particular point in time. My team has built about 100 validated signatures that we'll have a closer look. The methodology that we use to develop these RNA biomarkers for patient certification and selection is called Responder ID. We've built a foundation model of cancer biology that integrates the signatures that I mentioned before, and we can use this foundation model to map RNA-seq data into disease biology state, where we can then use background domain knowledge, like in this illustration, biology-informed filters, to infer the relevant phenotypes. I'll briefly explain the foundation model, and then we'll have a look at the use cases. Just simply put, right, what does it do? Our foundation model organizes cancer patients into clinically meaningful subgroups. We've heard a lot about large language models, LLMs. The foundation model that we've built is a large molecular model, LMM. So what it does is it maps RNA-seq molecular data into cancer biology space. So specifically, each patient's RNA-seq sample is characterized with about 100 scores, where each of the score corresponds to a particular biology signature. Let's take a closer look. We integrate specialized machine learning models where each of these machine learning models captures unique biology. I'll showcase the tumor microenvironment model in the following use case. A key component here is our universal space transformer. That's a deep neural network that adjusts the bias and adjusts the variance of RNA-seq data. And the output of our foundation model are 128 signature scores that I mentioned. Now, the second part of responder ID methodology is that we fine-tune this model to patient outcomes. We infer disease phenotypes based on biology, and then we link these disease phenotypes to therapies to make informed treatment decisions. The foundation model was trained on both public and proprietary data. We ensured global diversity by working with data providers or cancer centers such as Sidra Health in the Middle East, Academia Sinica in East Asia, 4Base Care and Cepian Biosciences in South Asia, and in the North America we work with Tempus, the Moffitt Cancer Center, and Exact Sciences. How we put this foundation model into practice. In the first example, I'll talk about Zerna TME panel, which was developed in collaboration with Oncology Therapeutics. The biomarker panel is now in prospective validation in clinical trials. The panel identifies dominant biology in tumor microenvironment. It predicts 4 subtypes that are characterized here as angiogenesis, immune suppressed, immune active, and immune depleted. You can also consider it on 2 phenotype axes based on this immune score and angio score. So both angiogenesis and immune suppressed phenotype would be high in abnormal blood vessels expression, right? And immune depleted and immune active would be low in these blood vessel expressions. The same goes for the immune axis. The TME panel workflow starts with processing tissue samples, right, and extracting RNA, and ends with this discrete phenotype prediction illustrated here in one of the 4 subtypes. We work typically with FFPE tissue samples from recession or biopsy. The RNA is extracted and sequenced and then analyzed by this universal space transformer, followed by a shallow neural network with 1 hidden layer and 2 neurons inside. We chose a simple architecture on purpose because it allows us to unbox it, as you'll see in the following example. The plot to the right is a representation of this latent space of the 2 hidden neurons that actually capture the 2 relevant biologies of the tumor microenvironment. We looked at the weights in this neural network. The genes that we selected were curated from the literature. Illustrated to the left is the immune phenotype, and we selected about 100 genes, some of which were related to the immune phenotype, others were related to angio phenotype. When you look at the model weights of the two neurons, you can see that for the first neuron, it assigns the neural network inferred that on its own, right? It assigned high weights to the immune genes. And the second neuron was assigned high weights to the angio genes. But you can also see some interplay between the two biologies because some of the genes are relevant for both neurons. If we look about the four phenotypes just to see how it matches our hypothesis, so we did gene set enrichment analysis to MSIG-DB signatures. The signatures were not, they did not overlap with our gene signature. And as it turns out, angio phenotype and immune suppressed phenotype is related or has high activation scores of angiogenesis and mesenchymal biology signatures. And in another example, when you look at the immune high phenotypes, which are immune active and immune suppressed, they have high activation scores for the inflammatory response signatures. You can also see that the immune suppressed phenotype, in one case, it's related to the angiogenic phenotype. In another case, right, it's also immune high, this immune suppressed, it's related to immune active phenotype. But really, when we explored it a bit further, we could confirm that this is indeed another separate, distinct phenotype from the two, and it's related to immune suppression. Again, here we're using MSIG-DB signatures that don't have overlapping genes with ours, and we see that this immune suppressed phenotype is related to immune suppression, in fact. That's how we unbox the machine learning, the neural network black box. So we can visualize the latent space. The x-axis is the output from the immune neuron, and the y-axis is the output from the angio neuron. The contours on this plot give us prediction probability that the sample belongs to one of the four phenotypes, as illustrated on this figure. When you observe or investigate data on this plot, right, you can make some treatment decisions. You would expect immune high patients to the right to be good fit for immune therapy. You would expect angio high patients to the top to be good fit for the anti-angiogenic agents, right? And the top right corner with high angio and high immune, that would be good fit for a combo therapy. In this case, we have gastric cancer patients that were selected for immune checkpoint inhibitor monotherapy. You can see most of the responders here to the right. The responders have green color. You can also see that response is the most enriched in the immune active phenotype to the bottom right. So there is where we have most of the responders, and if we were to choose this phenotype for patient selection, we would have a response rate of 58% compared to the standard of care that considers the PD-L1 biomarker and has a response rate of about 30%. When we look at the prediction probability of this biomarker, you can see it has bimodal distribution. It's a clear-cut separation between responders and non-responders. And when we compare that to traditional PD-L1 biomarker to the right, it just gives you better better choices for thresholding. We validated the panel under various conditions, in various tissues, for various therapies. In total, that's an old slide. It was validated on more than 10 cohorts and more than 5,000 patient samples under different disease modalities. We also looked at the The prospective capability, we did most of our analysis in gastric cancer patients, but when we look at other tissue types, in this case melanoma and colorectal, we see that it fits our expected hypothesis that immune-active patients have the best prognosis in this case. Just to wrap up this use case, the ZERNA biomarker now is in prospective clinical trial validation. What's also interesting to me is that it was developed originally on gastric cancer cohort, on microarray data, and now in clinical trials we have the same model but used in ovarian cancer and on RNA-seq panel. We'll just briefly look at the second case study for the sake of time, which is on KRAS-ID biomarker. We just launched this last month. The KRAS inhibitors burst into the scene last year. Treatment is assigned, as mentioned, based on the presence of absence of the mutation. The response rates are in the range of 30 to 40%. We can do much better when we look at the biology. Specifically, we looked at these 6 biology signatures listed on the slides, and those phenotypes were then included in the decision model. That's the overall schematic, just illustrating how the decision model for patient stratification is built on top of the foundation model, and it gives us response probability. When we take an inside look, right, this is how we could interpret individual predictions. On the slide, you see 2 patients in blue and in gold color. The patient in gold color would be predicted as responder just based on the biology, and the patient in blue, because of low activation of these biologies, would be directed towards alternative treatments. We did validation in preclinical data. Here I'm showing 2 of the axes, 2 of the biologies, because not all biologies are relevant in cell lines. They don't have the tumor microenvironment. You can see validation in 3 different drugs, 2 drug modalities, and when you look at the numbers, it gives you exceptional classification performance, AUC above 0.9 in all but one example here. We also partner with Tempus Therapeutics. We have real-world data of KRAS inhibitor treatment that was administered to G12C mutated samples. The black line shows selection based on the standard of care. We can do better, right? When we look at the complex biology, we can infer KRAS-ID high subgroup and KRAS-ID low subgroup. We can see that the KRAS-ID high subgroup has twice or doubles survival time. Power calculations indicate that when using this biomarker, you need 4 times fewer patients in clinical trials for the same power. And since we launched this biomarker, we started collaborations with several big pharma and medium pharma companies, including Roche, Revolution Medicine. Just to conclude, the regulatory environment is ready to consider biomarkers based on RNA-seq and based on machine learning, but it's important that these biomarkers are explained in detail, interpreted, and validated on independent data sets. Thank you.
Okay, we have time for a few questions, and I've remembered the iPad, so there's some questions from people online. Are these neurons in the model feature slide from foundation models or a fine-tuned model?
So the neurons are features from the foundation model.
Okay.
Okay, but then on top of that, we have another model that's fine-tuned to patient outcomes.
Okay. Sorry, not got my glasses. Sarah?
I was trying to get an idea as you were talking about this.
Sarah, could you mic just for people online?
Oh, sorry. Very interested in your biomarkers. You talked very little bit at the end there about the regulatory environment. And I was wondering, as we build these sorts of AI models for use in clinic, we obviously want to be able to update them. How is that going to work with the regulatory environment with such large data sets and constantly evolving ones?
That's a great question. The way it works today is when you update the model, you have to go through the whole process from start to finish. Through the Alliance for Artificial Intelligence in healthcare, we work with government bodies on policies, right? So we are proposing a framework where you could propose a model retraining protocol that models could be retrained under certain conditions. And then also in advance, you would also propose validation procedure for the updated model so that you could get the updated model through the regulatory faster.
Dave?
Thank you. Could you tell us a little bit more about that UST? I think that model to the left of the final one you were talking about. Thanks.
So what we did was we aligned or we removed the batch effect with methods like combat. You probably know combat, right? But combat is, so you have these population-based methods that can help you adjust the bias or variability between data sets. However, they are not really practical when you need to be able to map individual new samples into this universal space. Especially when you don't know the original bias modality, right? When maybe you get a cohort with just a few samples and you can't use traditional population-based methods for batch effect removal. So what we did was we invested a lot of time into building this aligned space of all the available RNA-seq data that we had of hundreds of thousands of RNA-seq samples. And then we trained a neural network that maps raw gene expression into this aligned space, into the space where the variability is removed.